So today we'll discuss about structure of kidney. Kidney is covered by one outermost fibrous capsule. The capsule also lines the renal sinus and forms tubular sheath or covering around minor calyces, major calyces, renal pelvis. Capsule can be easily separated during dissection. The cavity of the kidney is called actually hilus, where the blood vessels they enter, ureter comes out. That cavity is called actually hilus. For example, suppose this is a kidney. So each kidney has a cavity. That cavity is called actually what? So this cavity is called actually hilus. The hilus of the kidney opens into a space that is called renal sinus. So here you can see this whole area. So this whole area, dot area, I'm dotting this one. So this whole area is the actually renal sinus. The renal sinus contains actually what? Renal pelvis, funnel shaped expanded upper end of the ureter. If you see this one, so this is the ureter actually. This is the ureter. And the ureter is forming a funnel shaped expanded structure like this. This is called renal pelvis. And the pelvis actually what? Opens into the this renal sinus. We'll discuss. The renal pelvis divided into two major calyces. Singular is the calyx, cranial and caudal. Again, each major calyx further divides into several minor calyces. The renal sinus is filled with the fat actually. And calyces, pelvis, intrarenal blood vessels are embedded in this fat. We'll discuss actually. If you see this whole, so this is the ureter actually. This is the ureter. So this is the expanded part is the renal pelvis. It forms the calyx actually. So you can see this part. So we can see this part is the cranial major calyx and this part is the caudal. So this part is the caudal major calyx and this part is the cranial major calyx. Again, what if you see each major calyx has suppose this is the caudal major calyx. So you can see, so these are the minor calyx. So these are the minor calyx. Means each major calyx divides into several minor calyx. So this one is the minor calyx. And now this area actually filled with the fat. The sinuses filled with fat. We're discussing. So these are the sinuses. So here you will find the deposition of fat actually. So these areas, these sinuses are filled with the fat. So this open space. So this is called actually sinus. It is filled with the fat. Then kidney substance, or we call it as the parenchyma. The outer pale zone is called cortex. If you see, so this outer pale zone, so this is called actually what? It is called cortex. Outer pale zone. It contains convoluted tubules. We'll discuss, means PCT and DCT. Proximal convoluted tubule and distal convoluted tubule. It also contains blood vessels and lymph vessels. It also contains what? Malpighian corpuscles, means it contains Bowman capsules plus glomerular tuft. Then the inner dark zone is the medulla. So this is the cortex, but these are the inner dark zone. So these are the medulla actually. So medulla has 8 to 20 triangular masses called as renal pyramids, also called as the medullary pyramids. Their space is dependent actually. You can see, so this is the medulla. So you can see these triangular masses. Okay, you can see, so this triangular mass. So these are actually the renal pyramids. They're also called medullary pyramids. So these are the triangular masses. So their base actually faces the cortex. So this is the base area. So this is the base actually. This is the base of the pyramid. The base faces the cortex because this is the cortex. Okay. Apex. Okay, their apex is called actually renal papilla. Project into the minor calyx. So you can see that. So this is the apex actually. This is the apex of the each renal pyramid. Okay. And they project into what? So suppose this is the apex. This is the apex. Okay. Apex of the renal pyramid. It is called renal papilla. It project into what? It project into your minor calyx. So each pyramid contains what? It contains straight tubules, okay, of nephrons, blood vessels, lymphatics. But in case of small ruminants, like your goat and the sheep, you will find single renal papilla. Very, very important. In this diagram, we can clearly see. Okay, so you can see this structure is the renal pyramid. This is the renal pyramid. Okay, so this end actually, so this part I am shading with the green color. So this part is actually your what? Or this part, they are called actually renal papilla. You can see also renal papilla. So these are the renal papillas. Two renal papillae. Now they project into what? They project into this space. So this space, so they are called actually minor calyxes. Okay. So they are actually what? So these are actually minor calyxes. You can see minor calyx. Now they open into what? The major calyx. So these are the major calyx. Okay. And together they form, this is called renal pelvis. This is called renal pelvis, and this is called actually the ureter. Clear? So just we are discussing this one. The apex of each renal pyramid, that is called papilla, opens into a funnel shaped structure called minor calyx. Okay. So this is the renal papilla. Okay. Opens into what? The minor calyx. Minor calyces from the cranial part join to form major calyx. Okay. So these are the minor calyx. Okay. So they form the, so this is the cranial. So this part is the cranial major calyx. Similarly, minor calyces. So these are the minor calyces from the caudal part. Okay. Caudal part. These are actually, they join to form this structure, the caudal major calyx. Both major calyces merge to form the renal pelvis. So you can see. So this is the cranial. So this is the cranial major calyx. This is the caudal major calyx. Together they merge, and this part is called renal pelvis. Okay. And from that, what happened? Ureter arises. This is the ureter. Clear? Which marks the beginning of the ureter. A very, very important concept. Okay. Renal lobes and lobules. Actually what? A renal lobe consists of what? One pyramid plus overlying cortex. For example, this is the renal pyramid. Okay. This is the overlying cortex. So this whole structure, this one is called the renal lobe. Okay. Means one pyramid plus overlying cortex. So these lobulations or lobes are well marked in the cow. Okay. Not externally visible in small ruminants like dog, cat and horse. Again repeating. So these lobes or lobulation. Lobes is what? One renal pyramid and its overlying cortex that form the one renal lobe actually. Okay, for example, so suppose this is the renal pyramid. Okay, the overlying cortex area. Suppose this is the overlying cortex. So now this entire whole structure. So this whole structure is called one renal lobe. Okay. So these lobulations are well marked in the cow, not externally visible in small ruminants, also other animals like dogs, cat and horses. Each renal lobe is further subdivided into renal lobules. Okay. Lobe further subdivided into what? Lobules. Clear? Then we discuss about the blood supply of the kidney. So I will explain this with the help of one diagram. The blood supply to the kidney. The kidney will get blood from the abdominal aorta. Abdominal aorta gives the branches that is called renal artery. Okay, it will give the left and right renal artery. Now the renal artery will enter into the hilum or hilus of the kidney. So first they gives branches. They are called segmental branches. Okay. Then, so this is the renal artery. Okay. And these are the what? These are the segmental branches. Now, the segmental branches further form branches. Okay. So these are the branches of the segmental branches. So they are called interlobar artery. They further form branch. Okay. They further form branch. They are called arcuate artery. They further form branch. They further branch inside the kidney. They are called interlobular artery. Again, they further branch. That is called afferent arteriole. Okay. This afferent arteriole actually they form the your glomerulus. Clear? So now you can see. So these are actually the glomerulus. Okay. So this one is the actually your afferent arteriole. Okay. So this is the afferent arteriole. So the afferent arteriole. Okay. They form glomerulus, tuft. Then from that, this one will be the efferent arteriole. Okay. And now this capillary network you can see here. It is called peritubular capillary plexus actually. Clear? Again repeating. So now you can see this one. So this one is the afferent arteriole. It enters the Bowman capsule here. You can see this is the Bowman capsule. So now here they form a tuft of blood capillary that is called glomerulus. From this arises your this artery, this arteriole actually. This is called efferent arteriole. Then again after that they form a capillary network around the renal tubules. This is called peritubular capillary plexus. Nerve supply. The kidneys are supplied by renal plexus, autonomic innervation. Now we discuss about the nephron. Okay. So in this video, we'll only discuss the basic concept. Details you will study in the histology actually. All you know that nephron is the basic structural and functional unit of kidney. Each nephron consists of what? It consists of Bowman capsules. Okay. PCT, Hennell's loop and DCT. So first come to the Bowman capsule. Okay. So you can see it is a spherical structure Bowman capsule. Okay. A spherical structure that invaginates to enclose glomerular tuft capillaries from afferent arteriole. Okay. So you can see that this one, so this whole structure the Bowman capsule. Okay. So this outer layer actually is your what called? It is called actually parietal layer. It is called actually parietal layer, the parietal wall. And the inner wall, this is called actually your what? Visceral layer. All you know that. A double wall cavity. And here this is the tuft. This is the glomerulus. Clear? So the visceral layer, inner layer closely covers the glomerular capillaries. Parietal layer is the outer layer. It is made up of simple squamous epithelium and continues as the proximal convoluted tubules. So actually this parietal layer. So this तो ये वाला जो layer है. So this is the parietal layer actually. So the parietal layer continue as the PCT. So all you know that PCT, proximal convoluted tubule, longest and the most convoluted segment of the nephron. So this part. So this part is the PCT. Hennell's loop, a thin straight portion connecting PCT and DCT. So you can see that. So it composed of a descending limb. So this is the descending limb of the Hennell's loop. This is the U-shaped bend, and this is the ascending limb of the Hennell's loop. And this part is the DCT. Short and less convoluted than the PCT. Then the straight tube. Okay. The collecting tubule receives urine from the multiple nephrons. So this tube is the collecting tubule. Okay. The collecting tubule, they successively unite to form duct of Bellini or papillary duct, which opens into the renal papillae of each renal pyramid. Actually, you can see here, very important concept. So this is the one renal collecting duct. Okay. This is another collecting duct. So here another collecting duct will be there. So now all the collecting duct, they unite together and they form a large duct that is called duct of Bellini. Duct of Bellini. They are also called papillary duct. Now they actually they opens at the renal papilla of each renal pyramid. And many times you will find a concept that is called medullary ray actually. So a medullary ray is a group of straight tubules that extends from the medulla into the cortex of the kidney. Okay, this is the medulla. So from this you can see the straight tubules are radiating from medulla to the cortex. Okay, so the straight tubules actually they consist of straight parts of PCT, DCT and collecting ducts actually. Okay, so that's why they are called medullary ray. They radiate from medulla to cortex. They are the straight parts of PCT, DCT collecting ducts. They are collectively called as the medullary ray. Then now we discuss some species difference. Kidney of the horse. If you see shape, the very important, the right kidney is the heart shaped. You can see. So this is the right kidney, somewhat heart shaped, but the left is the as usual normal bean shape. Very important. So on the surface you will not find the lobules. Okay, so no lobulated, no lobulation, smooth outer surface. Okay, renal pyramids not prominent at all because lobes are not there and almost indistinct. Okay, very important point now. Okay, the renal pyramids are not distinct. Okay, so what you will find, the central part of the intrarenal medulla forms a concave ridge actually, and that ridge is known as what? Renal crest. Very important, renal crest. In this part you will not find the distinct renal pyramids. So instead of that you will find a concave ridge that is called actually renal crest. Okay, the renal crest projects into the renal pelvis and contains openings of the renal tubules. This perforated region is called area cribrosa. So you can see that. Okay, area cribrosa. Okay, this contains what? This contains openings of the renal tubules. So it forms a perforated region. Clear? That is called renal cribrosa. Kidney of the dog. Both kidneys are bean shaped. Surface smooth, outer surface not lobulated. Hilum renal pyramids. Hilum located at the middle of the medial border, relatively wide. Renal pyramids are fused, forming a crest similar to that of the horse. So here also we will find the renal crest. True pyramids do not exist. Okay. In transverse section, they appear unipyramidal, but in LS, long sections, multipyramidal due to the invagination of the pelvic processes. Kidney of the pig. The shape, if you see, both the kidneys are bean shaped, surface smooth, but some lobations are visible externally. Calyces, if you see, each kidney has 2 major calyces, around 12 minor calyces, approximately a dozen. Kidneys of the fowl, very important. The brown color, elongated structure you will find. is kidney. They present on the either side of the backbone, okay, vertebral column, below pelvis and sin sacrum. Surface is highly lobulated. Each kidney has three parts: the cranial part, middle part, and the caudal part. Now, just a brief review. Lobation and renal pyramid variations in the animals. Already we have discussed. In cattle and buffalo, you will find large units. You will find what? Lobes you will find. Okay. Kidneys are distinctly lobated. Number of lobes ranges from 18 to 22. Okay. So in case of cattle, buffalo, you will find the lobes. Okay. In birds, each of the three lobes is finely lobulated. Numerous small lobes are grouped together, give the kidney an irregular appearance. In humans and pigs, surface lobation not distinct. Each kidney has multiple pyramids. So you can see. So these are the multiple pyramids. But in case of goat, cat, rabbit, dog, no surface lobation, means you will not find any lobes on the surface. Each kidney has only one pyramid. Very, very important. These are unipyramidal kidneys, no minor calyces. So you can find only one pyramid. In some animals like horse and sheep, already we have discussed, renal crest you will find in place of pyramid. Okay, the crest forms due to the fusion of 10 to 20 renal pyramids.